One's kind of tucked away right underneath right here. This one has a nice coronal cut so we can see right into it. What are those called? There's valves that separate the atria from the ventricles, okay? And then there's the ones that separate the ventricles and they go at the base of the major arteries leaving. And I will say, because I haven't really went over this, so I'll put that's the aorta, okay? And that right there is the pulmonary trunk, but it actually gives rise to, I will say, the pulmonary arteries. So the pulmonary trunk goes like this, and then it splits off into the left and right pulmonary arteries. It goes like this. We just can't see the other one right there, okay? So what are those valves? So the ones that separate the atria from the ventricles are the AV valves. Atrioventricular valves. Yes? They separate the atria from the ventricles. So here's, we would call it the left AV valve. And actually we would say the tricuspid and the bicuspid, those are other names for them as well. Right ventricle is right there. Right atrium is right there. And then we have the other set of valves. What's the name of the other set of valves? That's at the base of the great arteries leaving and then separating from the ventricles. So right there. This is called the pulmonary semilunar, right? Pulmonary semilunar valve. And then this one, right, that goes up to here, I'll do it right down here. It's the aortic semilunar valve. Yes? A little crescent shape, you can kind of see it right there. You can see this right there. So there's 3 cusps and 2 cusps. It's kind of hard to see here, but do you see these little structures down here? We're going to look at these more later. 3 of them are there. 2 big fat ones are here. Those are kind of anchors that anchor those valve cusps down into the myometrium. And we're going to look at those in just a couple minutes, a little bit more detailed. Wow, that looks horrible, but yes, do those valves and chambers make sense? Okay, so that's the basics of the gross anatomy of the heart. So those valves are important because they're actually what allow the conduction of blood flow through the different chambers and vessels of the heart. And they keep blood moving in one direction. You don't want backflow of blood, and you don't want blood going in 2 different directions. You don't want merging of oxygenated blood versus deoxygenated blood. And this is maybe a good point to buzz out. I won't stop watering. It looks like I'm crying. Okay, so hard to see this, but artery versus a vein. So this terminology will come into play when we're talking about pulmonary and systemic circuits. What is the definition? Can someone tell me what's the definition of an artery? What is an artery? It's a blood vessel that, okay, so it's a blood vessel that carries oxygenated blood? Okay, that's what you might say. And that's what in general is the case, but it's not the best definition. Really, an artery is a blood vessel that carries blood. I heard someone say it.
Away.
There you go. Away, from the heart. Not all arteries carry oxygenated blood. Okay, not all of them do. It's not a definition of, oh, those are arteries, they're red. Oh, those are veins, they're bluish. No, that's not it. It's whether they carry blood in a direction that goes away from the heart, or, let's take the opposite of that. By that definition, then what is a vein? Yes. This is what carries blood back to the heart. Not whether it's oxygenated or deoxygenated. Because if you remember it as oxygenated, deoxygenated, when we look at the actual heart or we look at models in lab, you're going to see red or blue and go, artery, vein. Not always true. So leaving the heart, there are blood vessels that leave the heart and they go pick up the oxygen in the lungs. Those are arteries, but they're carrying, for the most part, weakly or low oxygenated blood because they're getting it at the lungs. They're going to get oxygen at the lungs. All right, so the valves keep that blood going in one direction, and they allow separation of deoxygenated versus oxygenated blood. So there's a fibrous skeleton of the heart that surrounds all four of those valves. The two semilunars and the two AV valves, the tricuspid and the bicuspid. It's dense connective tissue. So you looked at that tissue. The fibrous skeleton of the heart, it's basically like this core that surrounds those four valves and anchors them and protects them. It's made of dense connective tissue. What does it do? It actually anchors the valve cusps, because this is important to keep the blood flowing in the right direction, okay? Prevents overdilation of the valve openings, so that the valve openings, the cusps, they hang down and then they sort of close up, and then they kind of open, they close up. Prevents them from opening or dilating too wide, right? There's this skeleton that sort of anchors them, surrounds them, and prevents overdilation. And it's the main point of muscle insertion as well. So you've got the fibrous skeleton going around the valves, and then the muscle, the myocardium, kind of anchoring to it, surrounding around it. It also blocks the spread of electrical action potentials, or depolarization, that we're going to look at in a bit. How the heart actually has a series of electrical currents or action potentials that flow through it. It prevents, it kind of separates that. It allows that to occur in a very specific direction and sequence. So that fibrous skeleton is kind of a blockade to that. It allows the heart to kind of keep its atria and its ventricles and the conduction the way that it has to happen to make a nice normal beat. So if we look at this fibrous skeleton, it's dissected right out. If you do sort of a little transect right through, you can cut out that sort of fibrous skeleton that surrounds all four of those valves, okay? Looking at the semilunars and the tricuspid and the bicuspid. So if we look at that here, we've got the myocardium that anchors in, the muscle that anchors into this fibrous skeleton, and all those individual valves. So we've got the aortic valve, the pulmonary semilunar valve, aortic semilunar valve, the mitral or bicuspid or left AV valve. Okay, so that one has two valve cusps. And then we have the tricuspid or the right AV valve that actually has three cusps. Okay? So this one's over on this side. It's the right atria and ventricle separating. This one's over on the left side, separating the atria and the ventricle on the left. Okay? So this helps prevent spread of current in the wrong way, protects, sort of binds in and encases the valves, and allows muscle attachment. So it really helps to, the conduction of the heart and the way it's able to beat, it wouldn't be able to do so the way it does without the fibrous skeleton and the valves anchored the way they are. All right, so if we look at this piece by piece, the right upper chamber is the right border of the heart, and that's the right atrium. So we're starting up over on this side, okay, of the heart. It receives blood from the systemic circuit or the vena cava. The vena cava come from superior vena cava and inferior vena cava, and they dump into the right atrium. Okay? There's pectinate muscles that make up the atrium. These are ridges inside of the right atrium. And then there's a little structure that's left over from the beginning when I talked about development of that heart in the first month. There's a fossa ovalis in the adult heart, the child or adult heart. This is this depression that you can see inside, sort of right up against the interatrial septum. And at one point, this used to be, during development, a hole, a foramen ovale. Remember, fossa is depression. Remember the bone markings in the anatomical terms? And a foramen is a hole. So in the embryo and in the fetus, that development, there's a hole there. So basically, blood can go between the two atria. It's not critical to keep them separate. Blood is coming from placenta from mom. It's not going to lungs in a pulmonary circuit, so it's a different circuit. But in all of us, there's this little depression, this fossa ovalis that's left behind inside of the right atrium. If we go down to the right ventricle, this is getting blood from the right atrium. Okay, so it's coming from the right atrium, goes through that tricuspid valve, and during development, just developmentally, there's a change in the number of cusps. They both have sort of three on the right AV and left AV, but the right AV valve has its three cusps, whereas The left AV valve, two of the cusps kind of fuse together, so there's only two. So this one is the tricuspid valve. It's got the three remaining cusps that are part of the valve. It pumps blood into the pulmonary circuit through the pulmonary trunk. Remember that pulmonary trunk gives rise to the right and left pulmonary arteries, okay? And those go to the lungs to pick up oxygen as part of the pulmonary circuit, okay? The internal walls of the right ventricle are really kind of elaborate, and you can see their distinct structure. They have trabeculae carnae, they have papillary muscles, and they have chordae tendineae. Literally, papillary, like finger-like little muscle projections. They look like little fingers. Papilla is like a protruding finger-like projection. Chordae tendineae are, this translates to kind of heart tendons or cords, heartstrings. In Latin, this is like heartstrings. These are little tendons, little strings, basically, that attach to the base of the valve cusps, the ends of the cusps, and they anchor these down into the myocardium of the ventricles. Okay, so we'll look at what they look like in just a minute. And then there's the pulmonary semilunar valve. This is between the right ventricle and the pulmonary trunk. So the pulmonary SL or semilunar valve. All right, so those major vessels, and I kind of labeled them before, but the added structures that we just mentioned right here. So if we add, what is this valve right here again? Let's just do it again. This one right here. Right, AV valve. And another word for that is what? It has three cusps. Yes. Okay? So over on this side, this valve is what? Left AV valve, or it's got two valves, two cusps, the bicuspid. The structures that are in the myocardium wall, so you already know, right? That is the right atrium, and we're at the right ventricle right here. Those structures down the bottom, these are called the trabeculae carnae. These little anchoring at the bottom here. These are the little finger-like projections of muscle of the myocardium that pinch up and sort of look like little fingers. And I will take this over there. Papillary muscles. And then the little strings, basically they're like tendinous, sort of little ligaments, little heartstrings, little tendons that anchor the bottom of the cusps to the myocardium. What are those called? Yes. Chordae tendin. There's not enough room for me to write big and hunky on here. If you write small, you can do it. Chordae tendineae, papillary muscles. Notice there's three over here, two big ones over there. Trabeculae carnae, almost like the roots of the papillary muscles, like a tree. This is almost like the branches and leaves of a tree. The actual trunk of the tree is a papillary muscle. The roots of the tree that are embedded into the myocardium are the trabeculae carnae. You'll have to identify these in lab, and I expect you to know these terms, lecture and lab, right? Trabeculae carnae, papillary muscles, chordae tendineae. This helps to anchor the valve cusps, prevent them actually from everting the wrong way into the atria or the upper chambers. All right, now some of these other features, let's delete. Superior vena cava is bringing deoxygenated blood. It's a vein. It's dumping it into the right atrium. The right atrium carries that blood into the right ventricle, okay? The right ventricle pushes out through the pulmonary trunk and the pulmonary right and left arteries. That's the pulmonary semilunar valve that the blood actually has to push through. We also have the inferior vena cava that's bringing deoxygenated blood back to the heart itself. Right atrium, fossa ovalis is that depression that I talked about. It's an actual hole during embryonic development that connects the right and the left atria together. Pectinate muscles are the ridges or the muscle structure inside of the right atrium. Tricuspid valve, we talked about that. Right ventricle, that's the chamber. Chordae tendineae, papillary muscles, trabeculae carnae, pulmonary semilunar valve, that's where the blood leaves and goes to the pulmonary trunk. and then it goes out the left pulmonary artery and out the right pulmonary artery. Yes, so actually this was all filled in on yours, sorry, but it's good to actively sort of do it. Okay, but those are most of the names the structures that we just went through. Okay, left atrium. So now we're on the left side of the heart. We're over here now, okay? So the heart's posterior surface, basically is the back surface of the heart, receives oxygen-rich blood from the lungs via the pulmonary veins, okay? So coming back from the lungs, once oxygen is picked up, it comes back, it comes to the heart through the pulmonary veins. Notice these are rich oxygenated blood because they're returning from their lungs, going to the heart, so they're a vein. They're not bluish, right? They're not having oxygen poor blood, but oxygen-rich blood because they got picked up in the capillaries of the pulmonary circuit of the veins. So the left atrium opens in the left ventricle through the mitral valve. That's another term for the bicuspid or the left AV valve. Notice it has like 3 different names. A lot of surgeons and cardiologists will just call these the tricuspid and the mitral valve. Like they just call it tricuspid and mitral valve. They are the left and right AV valves though. Okay, so the left ventricle forms pretty much the entire apex of the heart. The left ventricle is very big. It's kind of sort of the biggest structure or chamber, strongest chamber of the heart. Very thick, strong muscle. So it has a really strong pumping action ability. The internal walls, again, have the trabeculae carinae, the papillary muscles, the chordae tendineae, just like, oops, I forgot, I have my drawing on. And it pumps blood into the systemic circuit through the aortic semilunar valve. So aortic semilunar valve is at the base of the aorta, boom, all the oxygenated blood goes out to the head and the rest of the body. All right, so if you fill in those, we've got the left atrium, the left pulmonary veins, here's the right pulmonary veins, pulmonary trunk, left and right pulmonary arteries, go to the lungs, pick up oxygenated lungs in the pulmonary circuit, then the actual veins that are returning oxygenated blood from the lungs are right here, and there's 2 of them, they're paired, and they dump right into the left atrium. You can see the little indentions right there, okay? So the left and right pulmonary veins. You have the mitral or bicuspid valve, which is the left AV valve. Again, you've got the left ventricle, the blood goes down into the chordae tendineae, the papillary muscles, and then the aortic valve. When this blood is leaving the left ventricle, it goes up to the semilunar aortic valve, which is right here, and then out the aorta. And the aorta actually is the ascending, the aortic arch, and the descending aorta, because it goes and then out to the different parts of the body, whether it's going down to the body or up to the neck and the head. Okay. Yes? So we got the valves, the structures, the internal, the external heart, and all the chambers. The walls of the heart differ in thickness. The atria are thin, the ventricles are very thick, especially the left. You'll notice in lab when we dissect the heart that the left ventricle actually is really thick, almost like steak. The right ventricle is kind of scrawny. You can cut right through it. If it looks real thick, I mean, so it looks like it's about 1 inch in certain sections, about 1 inch in thickness. It's very thick, it's very strong. The right is scrawnier, and the atria even scrawnier, even thinner. The systemic circuit is longer than the pulmonary circuit, so it has greater resistance to blood flow. It's longer, it's got more blood vessels, it's got longer way to go, it's got more capillaries and vessels that go all the way out, so it's got more resistance to blood flow than the pulmonary circuit does. Left ventricle, 3 times thicker than the right. So if we do a transverse cut right through the heart, you can see, here's the left ventricle, here's the right. And actually in a lot of ours, the right ventricle ends up squishing down into a little sort of crescent shape, and the left ventricle sort of takes up the bulk. Here's the apex. It takes up sort of the bulk of the ventricular part of the heart. So it exerts more pumping force. It's much stronger. Flattens the right ventricle into basically a little crescent. So it's when it's pumping and expanding, it's really the major pump of the heart. And notice the left ventricle, where is it sending blood? Where is blood going? Once it reaches, it's in the left ventricle, it's come in here, gone out to the lungs, come back, now it's in the left ventricle. Where does the left ventricle send that blood? To the body, into the head, right? Oxygenated blood, so it has to have good, strong force to get all that oxygenated blood out to the head and the body, okay? So, oops, you can just fill all that in on your own. This one I left blank, so you can make sure as you're going through, you can quiz yourself and make sure you get all those different vessels, chambers, valves, and structures. Okay. Cardiac muscle tissue itself, this is a recap because we learned histology at the beginning, right? Cells join are connected at intercalated discs, and we can see right on here, like there's a bunch of these intercalated discs where cardiac muscle fibers are anchored together. Complex junctions, they form cellular networks. They actually have protein that kind of links the adjacent branching cardiac fibers together, but they also have gap junctions. They have little holes or pores, little channels that allow ions like calcium or sodium and water to go back and forth between cells. So we looked at those and this structure of cardiac muscle under the microscope. If we look at these junctions, these intercalated discs, and we look real close, there's all kinds of fasciae adherens, these protein plaque sort of structures that on both sides, both of those adjacent cardiac cells, they're inside of those cells and up to the plasma membrane. They anchor, and there's little proteins that actually anchor the two separately together, and then anchor them inside of those cardiac muscle fibers. So basically they're stitched together and they're not going to rip apart. Do you want myocardial cells to rip apart? No. The heart is actually like a syncytium. It's this whole functional like connectedness of all these cardiac fibers, and they need to stay together tightly because their whole job is to pump as a mechanical pump, right? And they also want to share information so that they can spread that action potential from one cell to the next, and they can contract in a synchronized way. So there's gap junctions that allow ions and allow that depolarization of current to happen from one cell to the next. Okay, so gap junctions, fasciae adherens, that's what you find at the intercalated discs. Here's an intercalated disc, right? It's where those two cells are sort of linked up and connected together. They have sarcomeres, they have myosin fibers, they have actin fibers, so they have the myofilaments, they have the sarcoplasmic reticulum that stores calcium, that allows contraction or shortening. They actually do the sliding filament mechanism. You learned all that. It's the same sort of setup as in skeletal muscle. It's very, very similar. The overall morphology is a little different, but they do have bands. They have striations because of the thin and thick filaments, or the I bands and the A bands, making up the sarcomeres. So the same way, they're triggered to contract with calcium entering the sarcoplasm. They're cytoplasm of the myocardial fibers. Calcium signals the sarcoplasmic reticulum to release more calcium. Okay? So calcium comes in from the outside and triggers the SR to open up and release more calcium. So these cells depolarize, they get depolarization, and calcium comes in and then it triggers more calcium to be released. So we call this in physio calcium-induced calcium release. It's a really important part of heartbeat or heart contractions. The ions diffuse to the sarcomeres, so they go across, the calcium goes across all the sarcomeres. And then once you have all the calcium and all the sarcomeres, as long as you have ATP, remember, to allow the detachment of myosin, the recaulking, and then the attachment in power stroke, that whole mechanism is pretty much the same. Then you get contraction or shortening. Okay, so the fibers of the myocardium actually shorten, and that's what allows the atria to contract in series, in synchrony, and then the ventricles to contract in synchrony. So we'll sort of end right here, and then we'll come back to this when we start next time. What is a calcium flux trigger? So that calcium fluxing into the myocardial fibers, and what is the mechanism for that? Can cardiac muscle fibers regenerate? So if you damage cardiac muscle fibers somehow, lack of oxygen or totally damaged, can they regenerate? Can they regenerate and patch up and fix that area and regenerate new fibers? Provide a rationale for your answer. So you can even write these up. Think about this, write these down, and we'll come back to this when we have class on Friday. And that's when we'll finish cardiovascular and circulatory system. Okay? So if you could write those up and then we'll share those, we'll discuss them as a group. All right, so I'll go into the research stuff next time. And my thing didn't go off. Oh yeah, there it is. It's because I was recording.